Hey everyone, thank you very much for agreeing to give a Build a Cell seminar, and you can get started.
Okay, wonderful. Well, thanks, Kate, for the invitation. And as requested, I'm going to introduce myself a little bit. I think it's easier to share a slide. My name is Qiong Yang, and I'm currently an associate professor of biophysics at the University of Michigan, Ann Arbor. So I'm a physicist by training. I did my undergraduate research in phenomenological particle physics. And then I moved on to MIT to study the stochastic processes in single cell organisms. And I worked with Alexander van Oudenaarde. So that actually brings me into an interest in systems biology. So I went on to work with Jim Farrell at Stanford to study the robust cell cycles using the beautiful cell-free systems in Xenopus. And that also shaped what I'm doing now in my lab. So I integrate the tools of biophysics and bottom-up synthetic biology. And more recently, we got interested in mechanobiology. So I wanted to also have some discussion in this talk for the unpublished work. So to start my talk, I want to show you this movie here. Some of you might have seen it before. This is a huge Xenopus egg and that you can see by naked eyes. It's 1 millimeter in diameter. And after fertilization, it quickly cleaves into smaller and smaller cells in a high precision and synchronicity. So it really stands for the dream of every cell. And so the same process is well conserved across organisms. So here I'm showing you a zebrafish embryo. Because it's transparent, you can label nucleus and membrane by some fluorescence markers. And so what you see here is not only the cells are, the divisions are coordinated, but the downstream nuclear events are coordinated as well. So you may wonder if there are some timing system, like machinery that orchestrate all these complicated processes. And indeed, now we know that there is a clock. It's called a mitotic clock. And in this early embryo case, it's essentially the deposit molecules from the mother that interact to form these positive and negative feedback loops that can self-assemble into this self-sustained oscillations to drive the CDK1 on and off periodic activity. And the activation of the CDK1 and inactivation also coordinates the downstream nuclear events, such as the chromosome condensation, nuclear envelope breaks down, and spindle formation. So this is just a brief introduction about the mitotic clock. And indeed, the mitotic clock is just one of the many biological oscillators that span orders of magnitude in their time that controls various cellular and developmental processes. It's very important because if you interrupt of these clocks, it can lead to various diseases. So it's important for us to understand what maintains the clock function. But understanding it can be obscured by these complicated networks, which is typical for biological networks. It's like staring at the mysterious sky. We were wondering instead, can we apply bottom-up synthetic biology by asking, can we design a minimum oscillators? And by understanding these minimum designs, can we gain insights into the more complicated system? So we apply this synthetic biology in both computation and experimental systems. So computationally, we develop this framework. So here, instead of looking at one or two specific topologies, we look at the whole topology space. And in this case, it's a 3-node network that each node can be a protein or the gene that interact with each other with positive and negative interactions. And from this topology space, we could map into a functional space, such as understanding can these certain topologies do the oscillations, and are the oscillations robust or not robust, and can you tune the period and amplitude? So experimentally, we have applied on this Xenopus system by encapsulating the cell-free extracts into this water in oil emulsion droplets. And we recorded artificial cells. And inside, we can reconstitute the mitotic cell cycles. And we can develop the tools to track each individual cell so that we can quantitatively analyze the behavior of these droplets. And these are simple cells if you just include the cytosolic portion, and it can oscillate. But if you include the sperm DNA into the system, it actually self-assembles a nuclei that can be regulated by the clock itself, which is resembling the similar mechanism in the real cells. And additionally, we have developed this the CDK1 FRET sensor that allows us to measure the spatial temporal dynamics of the cyclin-dependent kinase activities. So to our interest is that, for example, in this view, you can see both droplets that without nuclei and some droplets that have nuclei. And both of these droplets can oscillate, but their oscillation profile is fundamentally different. And this is also interesting because it shows that this compartmentalization, such as the nuclear compartments, can play the role to affect the dynamics of these oscillations, which I will talk a little bit later. And so with the setup of the system, we could actually create thousands of droplets so that has a high throughput advantage. However, all of these droplets are identical in parameter space at the beginning. And so in order to really understand the topology to function linkage, we have to find a way to manipulate these topologies and being able to quantitatively measure the behavior after manipulation. So that brings this multi-channel design. So here we can have multiple channels and where you can inject different molecules of interest. And before when you create these droplets, that allows you to create a full dynamic range of certain molecule that you are interested to tune and in a continuous manner and in multi-dimension. So we think this is a random parameter scan in experiments. And that allows us to understand many questions that would be hard to understand in the intact live embryo system, such as how these cells cycles respond to different environmental perturbations, such as the free energy, the cytoplasmic density changes and nuclei to cytosol ratio changes, temperature, and also how the clock behavior, such as the tunability that I mentioned at the beginning. So I'm just to give a very brief example for the tunability here. So just like the signaling networks, the biological oscillators not only need to be robust, but also being able to take the input signal and processing it. And there seems to be different modes of this signaling processing. And one is amplitude modulation. And that has been shown in circadian clock, where it seems that when circadian clock is modulated, it's hard to tune its period, but its amplitude is more flexible to be varied by the external signals. But on the other hand, the cell cycle is the frequency modulated, and the frequency is easily adjusted by, for example, temperature and the nutrition that provided. And specifically for this CDK1 network, the synthesis rate of the cyclin B seems to be a input of the clock that can change the speed of these oscillators. But the amplitude maintains more or less the same. And indeed, this frequency modulation seems to be also shared by many other biological oscillators, such as heartbeats and neural spikes. And so we are interested to design experiments to really understand this frequency tunability. So here we are using a 2-channel tuning device. And because in theory, we know that the synthesis rate of cyclin B is an important input. And so we first tuned down this synthesis rate by adding the morpholinos of the xenopus cyclin, which will inhibit the xenopus cyclin B synthesis. And as we can see that this oscillation speed indeed is slowing down and eventually is arrested at some arrested state if you add a lot of the morpholinos. Now, additionally, on top of that, you can add human cyclin B mRNA, which is not affected by the xenopus cyclin B morpholinos. So we can rescue this, this oscillation behavior. And if you add more and more cyclin B mRNA, you see that the speed is increasing. And so because this is based on the FRET sensor, we can also measure the amplitude of these oscillations and allowing us to plot this amplitude to frequency relation. And we found it's a clearly frequency modulation where the period can change across a large dynamic range, but the amplitude is almost maintained stable. So this behavior, however, is depending on this the spatial property as well. So in fact, this tunability that we see can only observed in the cytoplasmic droplets, where you have a homogeneous spatial distribution of these molecules. If you now include the nuclei into the system, and that makes this tunability not being able to tune anymore. And in fact, the period is seems to maintain robust, independent of the cyclin B you add. And so that's still an open question in the lab. But here, for this talk, I want to switch the gear. So far we have discussed the single oscillators. And we know that the early embryos like xenopus is large in space. And even though each individual oscillators can be designed perfectly precise, how these oscillators coordinate from one end of the embryo to the other end is still a mystery. And diffusion alone cannot do the work because it takes hours to days for a molecule to diffuse from one end to the other end. And we know that our synchronized oscillations is taking about 15 to 25 minutes for different, depending on the species. So there has been an interesting proposal to suggest that there might be some wave propagation where the CDK1 can activate as a close to the centrosome and then propagating downwards. And in fact, if you look closer of these embryos that dividing, you find that the division furrow is not immediately it's actually it's going propagating from the animal pool to the vegetable pool. So to really understand this wave phenomena, there has been different wave mechanisms. So there are phase waves, which we also called it a pseudo wave, because this wave, like what I make an analogy here, like a wind structure, where each component here is not actually interacting with each other. And the fact that you can see the wave is only because there is a pre-established phase difference. And if you remove any component, it won't affect the rest of this behavior. However, for trigger wave, it's more like a domino effect. It's a chain reaction where one reaction is flipped the other. And so if you remove any component here, it will affect either terminate the waves or it makes the defacing of these waves. So about 10 years ago, in Jim Farrell's group, they reconstitute these mitotic waves in this 1D Teflon tubing. And they use the GFP attached to a nuclear localization signal to be able to observe the nuclei behavior. And that can in turn refer back to the CDK1 activity behavior. So in this case, they have a clever experiment where initially you see that there has been a wave that is observed in this chimograph, which is a space to time relation. And the red here is a mitotic entry, which indicated by nuclear envelope breaks down. And the blue here is the nuclear envelope, the nuclear reappearing, which is a mitotic exit. And so during the middle of the experiment, they cut the tube to create a physical barrier. And they found that the waves are actually shifted because of the cutting. So indicating that this is not a pseudo wave, this is actually a trigger wave. And so in fact, this wave phenomena has been important for not only biology, it's a crossing from physics to chemistry to this complicated biological system. And the scale also is diversified. It's essentially a self-organization, either at a molecule level or cellular level, or even at the organisms level. And it's important as well for the growth and the patterning of the embryos. So fortunately, even though the phenomena looks diversified, most of them seems to be eventually explained by this reaction diffusion mechanism. And depending on what kind of reactions, and you can create this bistable wave, excitable wave, or periodic wave. And so I wanted to highlight here is that it's actually important to have a pacemaker in space, where the pacemaker is defined somewhere of the space. The activity of this space is actually happening earlier than the rest of them. And therefore, by some diffusion reaction, there can be a wave that is propagating outward. So in our Xenopus system, we think that nuclei can behave as a pacemaker. Because as I showed you from the previous droplet experiments, we found that the nuclei, when you put in the system, in fact, the active CDK1 turns to translocate into nuclei. And that can effectively increase in the activity inside the nuclei and to promote an earlier mitotic entry, which will, and which in turn will trigger the wave outward. And so we want to go ahead and study how this wave is established. And to study that, we actually take advantage of this FRET sensor. And this actually allows us to directly observe this CDK1 spontaneous wave, we call it chemical waves, and independent of the presence of nuclei. So that would be a good control system for us. So I hope you can see that. So the top here is a one-dimensional Teflon tubing, and we injected a homogeneous the cytoplasmic extracts into the system. So at the beginning, because it's homogenized, everywhere it should be synchronized oscillations, and there is no or minimum heterogeneity. So what you see is that the tube across the tube everywhere is synchronized oscillation. But what's interesting is that at some point you start to see somewhere where the activity is dominating and propagating as a wave from one end to the other end. And so we use this chimograph to quantitatively understand the process. And as I just described at the beginning, across the space, everywhere is a synchronized oscillation, and there's no clear waves really at the beginning. But over time, you start to see this clear wave front, and we can analyze the slope to estimate the speed of the wave. And additionally, we can also use the peak location of the FRET ratio to indicate the period. And that gives us this sort of like a dispersion relation where we can see what's the speed in relation to the period of the oscillations. And it shows a very interesting time-dependent evolution. So at the beginning, we found there is a highly non-specified waves that has the speed all over the place, but it's also very fast speed. And we still trying to understand the nature of these waves. We call it the phase waves. And eventually they start to stabilize into this classical trigger wave regime. And it's this wave speed getting slowing down. So this is a phase to trigger transition. To really understand the transition nature, we collaborated with Dr. Lander Gallons and Danny, postdoc in his lab. And they built a model, it's a two-dimensional the partial differential model system. And so what they found here is that the synthesis of the second B, as I mentioned, which is important to drive the oscillator. And it seems that the heterogeneity of this the synthesis rate across the space is important for the transition. If you increase the heterogeneity, you start to get a faster and faster entrainment from the phase to trigger wave. So that means that if we had in the experimental systems to somehow tune the heterogeneity in space, we could manipulate the system. So that brings us to this experiment where we use the same cytoplasmic extracts, but here we add the sperm DNA into it and can self-organize into this nuclei. And so what we see here at the beginning, the nuclei here, if you zoom in, it can actually be observed because we can see the increased nuclear CDK1 activity in this case. And so at the beginning, these nuclei seems to enter into mitosis randomly. And this looks like a popcorn structures. But at some point, you start to see some nuclei in space is dominating and then propagating the waves. So that's sort of similar as a non-nuclear case. But if you plot this dispersion relation together, you find that the entrainment time in the nuclear case is much, much shorter. And so that is to showing that the nuclei in space, first of all, introduce heterogeneity in space, and second, that is helping the entrainment of this wave. And so then we wonder, can we actually have some external source as an entrainment system? So here, to test the idea, we started to use this CSF, which is a stably steady state extracts where CDK1 is arrested at a high state. And then we use, in the tube, we loaded the interface extracts where the CDK1 is stably low. So now if we dip the tube into this reservoir of CSF, it should have some initial activations at the end of this tube. And now we can remove the tube from the reservoir. And we found there is actually a bistable wave that is developing. And you can see a one single wave that is propagating from left to right. So that is to show that this dipping experiment is working. So we are using the extracts, the secondary extracts with and without nuclei. And for both, we found that there is immediate entrainment of these waves from the boundary. And so as a quick summary here, essentially we have look at the time evolution of this system. And essentially there is a phase to trigger transition, even happening in the spontaneous case where in the control case, you don't have an initial pacemaker. And however, if you add the nuclei or if you add some external source to entrain the system, both of them can help with the entraining of the system to be entrained into the trigger wave in the faster case. And with both of them, that is entrained the most. But what's also interesting is that even though these nuclei and the CSF dipping, which both are increasing heterogeneity of the space, can help entrainment of the the waves from phase to trigger wave, but the kinematic property of the wave is not affected by heterogeneity. As we see that the terminal speed is always stable about 40 minutes, 40 micron per minute. And that is even true for the bistable waves. So I think we still have some time. I want to also introduce some new work in our lab. And this is to look at the zebrafish growth and patterning. And specifically, we are interested in a developmental process called somatogenesis. And so somatogenesis is just the periodic formation of the somites, which is a precursor of the body segments for all the vertebrates. And in fact, for zebrafish and all the way up to our human, this process is highly conserved. And so the process happening where you have this single negative feedback loop called, that is formed by a HER1 gene that is producing the protein, and the dimerization of the protein can be repressor of the gene itself. So this is a single negative feedback loop that can allows this clock called the segmentation clock that oscillates. And what's interesting is segmentation clock and the mitotic clock is actually co-regulating the process, because at each somite formation, you also need an extension of the tail so that you can keep adding this the body mass. And so in zebrafish, the process of the somite formation is about 25 minutes per somite. So this is a very complicated process. And in fact, the current model in this field, the understanding is that it's a three-tier model, where, as I said, there is at the bottom, there is a single cell oscillator, which is a single negative feedback loop. These oscillators actually can synchronize by the cell-cell communications through notch delta signaling. And additionally, to provide some cues of the space, there is also the signaling gradients of the RA and FGF and Wnt that is oppositely reducing from the anterior and to from the posterior towards the other end. And so all of these seems to be important to shape this formation of the somites. However, additionally, especially recently, there has been study to show that not only is there is a chemical gradients across the tail, but the physical properties such as the cell motility, the cell density is also having showing a gradient across the tail bud from the posterior to anterior. And also there has been a measurement for the rigidity of the system using these ferrofluid droplets in the magnetic field. And they show that the zebrafish tail bud seems to be soft as a posterior and getting more stringent, more rigid as an anterior. So in our lab, we are interested to understand how the mechanical signals may affect this process. And because it's complicated to study in the in vivo system, we have the same strategy to dissect the system and use a single cell assays, trying to decouple these effects. And here we are developing this, dissociating the PSM cells from the tail bud, and then we can culture them on the different environmental, the mechanical environments. And here, in one, we put them on this glass surface, but with a pluronic coating. So the pluronic avoid the cells to attach to the surface to sense this the surface. So it's a slippery surface here, as you can imagine. And on the other hand, we have the matrigel coating where cells can grab it. And so what we see here is on the left here, hope you can see the video clearly. So we can see these isolated cells can do the oscillations because each of them has this single negative feedback loop. to allow them to oscillate. So about 50% of these cells can oscillate in this condition. However, the same cells, the same group batch of the cells, if you plate on a matrigel coated surface, you find these cells actually is spreading out, and they and most of them actually do not oscillate. And if you focus on the black arrows here, these are the cells that do not oscillate, and they also tends to migrate a lot. And so, and in fact, we have found that across different experiments, only 4% of these cells can oscillate on the matrigel condition. And so look closer, we can have the single cell trackings, and just as examples of this, left side is the pluronic coated cells, and on the right side is the matrigel coated cells. And what's interesting is the pluronic coated cells seems to be more or less round shaped, and the matrigel coated cells have the polarized shape. So we went ahead and to quantify using the circularity. And the oscillating cells, as we just see, has a high circularity, but the non-oscillating cells eventually it's polarized, and it also shows a high migration behavior, as we can see here. So on the left side of this figure, it's the oscillating and the non-oscillating cells. On the pluronic coated glass. As I said, the pluronic glass, the cells essentially do not attach into surface. So they maintain the round shape regardless of the oscillating and the non-oscillating. However, in the matrigel coated glass, these non-oscillating cells, when they are polarized, it also shows high MSD. And so, and this actually indicating it might be relating to some mechanical signal that the cells can sense and eventually talk down to the at the genome level. So there has been a study in mouse TSM cells, and they found a similar results where here they found that the when they put the cells on the BSA coated, which is similar to pluronic coated, the YAP is inactivated. And if they put the cells on the fibronectin coated and the cells spread, the YAP is activated. And that seems also correlated with the oscillating behavior. However, for both our studies and this study, we are putting the cells on the glass, and the glass has an extremely high rigidity, which is outside of the physiological range. And in our measurements using AFM, we found the rigidity of the zebrafish tail bud is about 0.67 kPa, which is significantly smaller than glass, which usually has a 50 GPa. And so with that, we wanted to build a system to actually have a dynamic range of the surface rigidity we can engineer. So we set up a collaboration with our colleague, Jianping Fu from mechanical engineering. And here we are using the micro-post arrays. And based on the geometry of the post, we can quantitatively manipulating the post rigidity. And then we place our cells onto this post array system. And to our interest, we found that this cell oscillatory behavior responding to the surface rigidity in a switch-like manner. And so, and we are also comparing with the pluronic coated glass surface and also matrigel glass. And essentially, the cells maintain a high oscillatory percentage as a soft surface. And at some point, about a 10 kPa orders of magnitude, they start to switch off and has presenting a low oscillation ability and comparable to matrigel glass. The cycle number shows a similar behavior. So that makes us interested on what might cause this behavior. Although it's still an open question, I think our lab still trying to explore more of this direction. But there has been a study to show that the YAP signaling actually is sensitive to the surface rigidity in the way that if a cell put on a soft surface versus the hard surface, the cells at the hard surface, it's first polarized as we see as well, but also it's causing the stretching of the nuclear pore structure, which allows the YAP to be more easily translocated into nuclei. And the measurement to show that there seems to be a switch-like nuclei to cytosol ratio in response to surface rigidity. So we built a phenomenological model taking into account of this effect of YAP translocation, and that can recapitulate roughly about this switch-like oscillation behavior. And if we look closer of these cells on the micropose system, we can also use how the cells can bend the post to estimate the traction force of the cells. And what we found that the oscillating cells, they are round shape. They also presenting a consistent low contractility, but the cells that are non-oscillating, when they polarize and migrate, they tend to grab the post more, and they show the high contractility. And then moving forward, we are hoping to building from bottom up. So trying to re-aggregate the cells and build a two-dimensional patterns by microcontact printing. And additionally, we have collaborated with Jianping's lab to build this somatogenesis model in vitro by the human pluripotent stem cells. And when we seeded these cells in this microfluidic system where we can have signaling gradient in space for both anterior, posterior, and also dorsal ventral, we can actually recapitulate this somate formation in these microfluidic chambers. And that can allow us to understand more quantitatively of this system. And additionally, my lab recently established a Pascois model to understand a very interesting behavior. It's called a wetting dewetting. And when you isolate this, the cells as a 256 cell stage of the embryo and plate on a surface, and they start to spread out and then de-wet. And this is actually resembling the process during the epiboly. So we are hoping also to explore more of this mechanobiology of this process. So with that, I want to thank the fantastic students and the postdocs in my lab. And the highlighted here are the people behind the work that I'm presenting today, and also my collaborators, especially Danny, Leonard, and Jianping, for this work that I'm presenting. And I think I should stop here and answer the questions.
Thank you very much. That was an awesome talk. I really like the visuals. There are some questions in chat. Can you see chat?
Oh, yes.
If you could read the question out loud before you answer, that's going to be for the recording.
Certainly. Let me see. So the first question is, so in your CSF bistable wave experiments, is the deeping experiment diffusion limited? And then is the wave amplitude depending on diffusion speed, so could be controlled by temperature? That's a very interesting question. So the first question here is deeping experiment diffusion limited. So we haven't tested the diffusion or manipulate the diffusion yet, but in our experience, the dipping cannot be very long. And in fact, we usually limited our dipping time within a few seconds. Otherwise, the whole tube is just ruined. And so it's also an indication that so actually, I probably don't have the slide there. In fact, we can measure the diffusion. Let me see if I can find the slide. Sorry. So here you can see that there is a hyperbolic shape during the dipping for both non-sperm DNA and with sperm DNA. And that is an indication of the diffusion of the molecules. And beyond that, this is the trigger waves. And so I hope that answers part of your question. And let me see your 1 second. Is a wave amplitude dependent on diffusion speed. So in fact, the amplitude of the waves, which is similar, if I can understand, to the oscillation amplitude of the CDK1. And that is making it very stable. And it's because of this nature of the clock design. There is a double positive and double negative feedback loop that controls the CDK1 activation from off to on. And the off, it's a bistable switch system. And it seems that the on state and off state, the distance is always keep the same, and regardless of the speed and other things. And in terms of the temperature, actually, we have a study right now to understand how cell cycle responds to temperature. We also study some wave behavior as well. So far, we don't have a clear conclusion yet, but I hope to update once we have a more interesting result. And so the next question, great talk, thank you for presenting. Is there a way to reproduce a cellular oscillator like you discussed, but in vitro? So I'm trying to understand so reproduce a cellular oscillator. I don't know, this is from Ben Jinsky. Do you mind if you clarify what kind of cellular oscillator in your mind?
If you want to unmute yourself, you can, or just type in chat.
You can, yeah, you can type in chat or you can, yeah, I think it's probably easier if you are comfortable to unmute.
Or we can move on to the next question and see if he clarifies.
Right, yeah. So with pure chemistry or perhaps with cell-free protein expression. So this is actually interesting because we were wondering as well for the actually truly bottom-up synthetic oscillator. So we have been trying, it is very challenging in cell cycles because even though the skipped down version, as I showed here for the xenopus extract, is still very complicated. The CDK1 and APCC, which is the anaphase promoting complex, both of them have multiple so both of them are huge complex proteins, have many subunits. So it's hard to purify, but I think it's interesting still and worthwhile to do to put them together in test tube and then encapsulate in the droplets to see whether it's actually working from bottom up. And as a more probably workable system is a Chi-ABC system in the circadian clock, if that's what you are asking. So in fact, there has been a study for building this clock in test tube just with the Chi-A, Chi-B, and Chi-C protein and with ATP. And they can oscillate with the circadian clock time as well as temperature compensation. So I would be interested to see how people can encapsulate that system in the droplet. So the next question, do we know what genes are responsible for oscillation of the zebrafish embryos cells. Yes, so there for zebrafish, there has been, if you're asking about the segmentation clock, it's a HER1 and HES7, and each of them can form their own negative feedback loop by dimerize themselves and then repress themselves. But they also have heterodimers from HES1, HES7, and HER1. So these are single negative feedback loops that are controlling the oscillation for segmentation clock in zebrafish cells. If you're asking the early embryo, like cleavages is the same as xenopus, it's a CDK1 network for controlling the cell cycles. And could you reconstitute that circadian other cell type and make them oscillate? So depending on what kind of cell type, so because for both cell cycle and segmentation clock, they're well conserved. So that means across different species, they are there. However, if you say, for example, can you engineer some Chi-ABC system put in a bacterial cell where they don't naturally have the system? And I believe the answer is yes, because there has been study to, if I remember correctly, to engineer the system to allow a normal like a bacterial cells to gain the circadian oscillations. And so,
So you have one question. Jan is raising his hand. If you want to unmute yourself and ask in person.
Yes, please.
Hello, hi. Can you hear me?
Yes.
Okay, perfect. Thanks so much for the amazing talk. I really enjoyed it. I had a question regarding this slide exactly, and this work. So I understand that the cytoplasm, that the cell extract is static, right? So it's not moving at all. It's like complete. You're letting the diffusion just take place in a static environment.
Sorry, could you repeat so are you mentioning about here?
In this just A. So basically the wave is moving through the cytoplasmic extract, and the extract is not moving at all.
Oh yeah, no, yeah. So
Yeah.
yes, it's exactly the idea of the flipping the bistable wave. So
Exactly.
on activity, yeah, the extract is not moving. Yeah, thanks.
Because I was just wondering if like obviously this is like a follow-up thing, but I was just listening to you talk and I just thought like if you had like a laminar flow, could you also have this type of mitotic wave, but like trying to have some form of cytoplasmic streaming, at least like, or would that completely mix everything up literally and get rid of the whole thing?
It's an interesting thought. I know in Drosophila system, people have studied this essentially as what you said, if there is some laminar flow, right? And sort of as a way to, instead of making it a heterogeneous, it's remixing everything, right? So that would be interesting. I think that depends on the time scale as well.
Yeah.
Comparing how this time scale of entrainment versus the other to remixing. But it's a, yeah.
Because the reason why I'm asking this is there's this organism, Physarum polycephalum slime mold. I don't know if you're familiar with it.
I heard.
Yeah. Oh, okay, yeah. So it's basically just like one huge cell. It's like a cell that can have 50 centimeters long. And the weird thing about it is that like it mixes all of its cytoplasm and it goes back and forth, and then it stops, and all the nuclei undergo mitosis at the same time, and then they start cycling again. So I was just thinking if what you're showing could maybe relate to this mystery of what is happening. Why does mitosis happen so in Physarum like that? And that what you're showing is a really nice. I mean, it might apply to this.
I would love, is there any public, I would actually love to learn about that slime mold system, because what you described, I'm not familiar with and I wonder why there is this mechanism going back and forth and then everything divided at the same time.
Yeah, exactly. Like so yeah. I was just wondering maybe you're familiar with this and this is like some area that you're building to. But I really loved your talk. Thanks so much.
Thank you for your question.
And I think we got clarification from Ben on his question.
Yes, thanks, Ben. So these are single cell oscillation. You show the second half. Yes, this is for zebrafish cell embryo oscillators. This is related to genetic oscillations. So that means the extracts needs to have a way for the transcription and translation, which I think is possible. And I believe the clock itself is simple enough. It would be interesting to test the idea. I think no one has tried, but I think based on the design of the clock, it's probably possible. Yeah.
Sounds great. If there are no more questions, I think all my questions have been answered too. And I just want to say thank you so much again for the talk. And thanks everyone for the great discussion.
Thank you so much.
And I hope you all have a good week.
Have a good summer.
And a good summer. All right, stay cool or warm or whatever, wherever you are. And bye everyone.